So the simulation you just did is basically following these steps that you see on this slide. So we know that before a cell can divide, before a cell can go through mitosis, the first step that needs to happen is the DNA needs to copy itself. In order to make two identical cells, you need a full copy of the DNA for each of them. And what you did in that simulation shows you how that happens. Because DNA is made of those complementary bases, what happens is the strand of DNA unzips between the bases. Those hydrogen bonds that connect the bases, they're pretty weak. So they can sort of just split up. An enzyme splits them in half, and then you have two sides of the DNA molecule. And then, just like you did, enzymes help to connect the complementary bases to each side. And when you finish doing that in the simulation, what were you left with when you were done? What did you create? And how did it compare?
About the same.
It was exactly the same, right? Because those bases are complementary, when it split and new ones were filled in, then you had two exact copies. They looked exactly, they had the same order of those bases as the one you originally started with. So that's how DNA replicates. It splits, and new bases fill in on each side, and you're left with two identical DNA molecules. So we're going to move on. So our DNA, as we just heard, we have a full set of all of our DNA in basically every one of our cells. Every cell has all of the information. It's not like a brain cell has a different set of DNA than your skin cell, which has a different set from a stomach cell. Basically, every cell has an exact copy of all of the DNA. And different parts of that DNA are turned on or off depending on what is needed by that type of cell. So a certain set of genes are turned on in your bones. That's different than the genes that are turned on in your cheek cell, which is different from the set of those genes that are turned on in your brain cell. Depending on where they're located, the different types of genes are turned on or off. And so how does DNA, which is the series of A's and T's and G's and C's, how does that give us our eye color? How does that give us our traits and our characteristics? What has to happen is that code in DNA needs to sort of be implemented and needs to do things in our body. And that process is what we're talking about now. So DNA on its own is basically stuck inside of the nucleus. It's sort of stored away there safe. In order to actually do its job and produce traits and characteristics, it needs another type of molecule called RNA. So DNA is deoxyribonucleic acid. RNA is ribonucleic acid. And it helps this process to happen. Really, it's the RNA that does most of the work in the cell to implement the genetic code. Mm-hmm. There's some differences between RNA and DNA. Like we just talked about, DNA is a double helix. It's double-stranded. There's two strands to it. RNA is not. RNA only has a single strand. Okay? It's set up in much the same way. It's not a double helix. It's a single helix. And it has mostly the same parts. It has a phosphate group, just like DNA. It has a sugar, just like DNA. The sugar is a little bit different. It's ribose. And then it has a base, just like DNA. And the bases it has are similar, but there's one difference. So RNA has cytosine, it has guanine, it has adenine, but it doesn't have thymine, doesn't have the T. Instead, it has a different base. It's called uracil. We abbreviate it with a U. And then that's the difference in the bases. And so we see here an example of an RNA molecule. We see the sugar phosphate backbone. We see the bases in between. There's cytosine, guanine, adenine, and uracil. And there's 3 different types of this molecule, each of which has a specific job to do to help the DNA do its job. We abbreviate each of the types of RNA with a letter. There's mRNA, which stands for messenger RNA. There's transfer RNA, we abbreviate with a T. And then there's ribosomal RNA. And basically all of these different types of RNA are helping to make proteins. When we really think about what the DNA does, it has the instructions about how and when to build different proteins in an organism. That's what gives us our trait. Why are our eyes the color they are? Because of these proteins that are pigments in our eyes. Why is our skin the color that it is? There are proteins, that melanin, that gives our skin its color, and there are genes that tell how much of it to make. Why do we have certain hormones like insulin? Our DNA has the instructions about how to make that protein insulin and when to make it. And so these RNA molecules help the process. Okay? They each have a job to do. The messenger RNA carries the information from the DNA out to the ribosomes. The ribosomes are the machinery that puts the protein together, and transfer RNA brings in the correct amino acids that are going to get linked together. So if we're comparing DNA and RNA, some differences, right? DNA is double-stranded, has 2 strands. RNA is not, just has 1 strand. The sugar is different, slightly. In DNA, it's deoxyribose. In RNA, it's called ribose. The bases are similar, but not exactly the same. DNA has A, C, T, and G. RNA has A, C, G, and U. They're the same in that they're both made of nucleotides linked together, and they have the same basic components. So let's talk about how this happens. So our genetic code, we already have said, it's the sequence of bases we each have. That's what gives us our traits. We have about 3 billion of those letters that makes up our genetic code in each of our cells. The DNA and the RNA have to work together to actually build proteins. So here's how it works. First of all, the DNA you know is in the nucleus. It doesn't leave the nucleus. DNA is this huge molecule. It's unable to get out of the nucleus. The nucleus does have, the nuclear membrane has little holes in it. Some things can move through it, but not DNA. It's just too large of a molecule. It's stuck inside of the nucleus. So when the DNA code is going to be used, a set of things happens. First, the DNA unzips in a certain area. And then the gene that's being produced is copied from DNA to messenger RNA. Messenger RNA is a much smaller molecule. It's only a single gene. It's single-stranded. Messenger RNA is able to get out of the nucleus. Which cell part is actually responsible for making proteins? We learned back when we studied cells. What's the name of the cell structure that makes proteins? Ribosomes. ribs. Remember ribs, protein, ribosomes? Yeah. So the messenger RNA that was just copied in the nucleus goes out of the nucleus and goes and finds the ribosome. Attaches to the ribosome. And then the messenger RNA has the code that says which amino acids are going to be strung together to form a protein. And so transfer RNA molecules start bringing in the correct amino acids. The ribosome links them together until the protein is made, and then it can be released. So it's easier to see this sort of in a diagram. Here's the two parts. First, this is in the nucleus. We see our DNA strand here. An enzyme comes in, unzips a part of the DNA, and then it starts to be copied into this messenger RNA strand. That's what this red one is. Eventually, the whole gene is copied to messenger RNA, and then it can be released. So if the DNA, for example, if let's say this part of the DNA has this sequence, GGCAATACCAGAT, what happens is the messenger RNA is built based on that. So if the DNA has a G, what does the messenger RNA have?
C.
C. G goes with? C with? Be careful. A? U. Because remember, RNA doesn't have the T. It just has the substitute. It has a U. And so eventually, the DNA is copied to this messenger RNA. It can get released and can go do its thing. That whole process of copying DNA to an RNA is called transcription. And then that RNA can go do its thing. It goes out of the nucleus. Its job is to go to the ribosome. Every 3 letters on the messenger RNA is called a codon. And it's a code to produce a single amino acid, or to attach an amino acid. AAU is one code. GGC is a different one. And CAC is a different one. Each of those 3 letters is a code for a specific amino acid. And when the messenger RNA is attached to the ribosome, the transfer RNA starts bringing in those amino acids. And the ribosome starts linking them together. There's a couple special codons. A start codon that says, okay, let's start building the protein here. And there's a stop codon that says, okay, we're done. Protein is finished. It can be released. This chart helps you to decipher which 3 letters correspond to which amino acids. The way you use this chart, let's say you were given a codon, AAG. To use this chart, you take the first letter, here it is, A. Then you look up at the top. A, what did I say? AG? A, see where they connect? It's in this box. And then G, AAG is a codon. That's a code for the amino acid lysine, LYS. And so you can use this chart that helps you to figure out what codon matches with which amino acid. So let's look at another diagram. Kind of shows us all of this. In the cell, in the nucleus, we have the DNA. It has all of the instructions about how to build the protein, but it's stuck there. A section of it unwinds, and it's copied to messenger RNA, which leaves the nucleus, goes out to the ribosome, where the code on the messenger RNA tells which amino acids should be put into making this protein. And they're brought in by transfer RNA one after another, and they can link together one after the other, making this long chain of amino acids. Here's another example. Okay, DNA copied to RNA, goes out to the ribosome. Transfer RNAs start bringing in these amino acids, which get linked together until the protein is finished. And then this protein can go do its job. It can be an eye color pigment. It could be insulin. It could be growth hormone. It could be muscle cell. It could be all sorts of different things. Those proteins are what give everything its traits. Here's another example. So here we have an animation. First, transcription. DNA copied to messenger RNA. Second step, translation. Transfer RNAs are bringing in different amino acids to get linked together to form a protein. Let's look at it again. First, DNA to messenger RNA. Then, messenger RNA goes to the ribosome. Transfer RNAs bring in all of those correct amino acids until the protein is finished. So that's how